Hello everyone, I'm Keng Fu Lan from West China School of Stomatology Implantology Department in China. Today I'm going to talk about identifying type 2 collagen alpha 1 chain as a novel therapeutic target for osteoporosis through bioinformatic integration. First, I will give a brief introduction about osteoporosis. It is the third most common disorder in worldwide, and it will reduce the bone mass and increase the bone fracture risk. And for our current treatment, the clinician prefer therapeutic or romosozumab as a prescription. Next, what's collagen type 2 alpha 1 chain? It's a gene that encodes type 2 collagen, usually can be found in cartilage and in eyes. And I will give a brief introduction about two most common omics, including single-cell RNA sequencing and bark RNA sequencing. The difference between these two is bark RNA sequencing can only show the sample's gene expression's average value, but it cannot separate it into different cell type. However, the single-cell RNA sequencing can compensate that disadvantage, but it is a very expensive and will require high-quality samples technique. So in our study, we try to combine these two. And first, we use the bark RNA sequencing dataset, one from the GEO database and the other from the bioproject database. And both experiment they used 8 weeks of reactomized mice and compare with the control mice. They used the femur head bone sample for sequencing, and in the GEO data sets, they separated cortical bone and trabecular bones into two results. So in the Venn diagram, we try to combine those two results into one and compare it with the bioproject results. And we can find there's a 73 common differential expression genes. Later, we run the 73 genes into the stream database to see the protein-protein interaction network and identify the hub genes. The hub genes including three hub genes, it's MYBPC1, DNTT, and CHAD. And next, we collect the two datasets from GEO database with a single-cell RNA sequencing data. Those both methods use the osteoporosis and non-osteoporosis patients' femur head bone samples, and we integrated two datasets with the CCA integration to remove the batch effects. And then the cell type composition, we can see that osteoporosis group had higher urocytes and adipocytes and had a lower macrophage and neutrophil. Moreover, that stromal cell will increase in osteoporosis group. However, stromal cell consists various of cell type, including chondrocyte, osteoblasts, osteocytes, stem cell, act. So if we want to see what's the proportional change in stromal cell cluster, we subset the stromal cell from the data. As we can see that osteoporosis group stromal cells is usually shown at upper part of the UMAP, and in the control group it's at the lower part of the UMAP. And for COL2A1 and ACAN, these two genes are highly associated with chondrocytes. It appears expression level is higher in the control group. However, that the COL1A1 and LPL is highly associated with osteoblasts, which is expressed higher in osteoporosis group. Later, we tried to analyze the different expression genes we found in stromal cells and compare with our previous bark RNA sequencing results, and we can see there's a 5 gene that is associated with osteoporosis, and 4 out of 5 were in the CHAD module. So we seek to see the expression level of these 4 genes, and those 4 genes express higher in the control group compared to the osteoporosis group, especially the COL2A1. And we want to confirm what's those genes expression in which type of tissues. We use the Human Protein Atlas database to confirm that COL2A1 and ACAN were found in human cartilage. But it doesn't mean that it's linked to the treatment of osteoporosis treatment. So we collect another dataset from GEO database with a therapeutic treated versus non-treated data. And we can see from the A, it's a UMAP of the data. And in B, we can see that COL2A1 and CHAD expression level is higher after therapeutic treated. Afterward, we also analyze it with a cell type composition, and we can see that therapeutic treated groups show higher macrophage, neutrophil, and chondrocyte, which contradict from the osteoporosis patient we get the data before. And below is the tools we use for analysis. In summary, that we use the different type of omics to confirm that COL2A1 is highly associated with osteoporosis. And to confirm our hypotheses, we also collect the treatment like teriparatide as our data set to confirm our hypotheses. In the end, thank you for you all listening and hope you have a good day.